her phone to sit on top of this contraption and basically FaceTime the whole ostrich parade so I could watch it.
That's hilarious.
Yeah.
All right, so we're back here. We're going to start with the cell again. You're not going to be able to see that on the video, but we'll try. So what's a cell made up of? What's the important stuff in here?
There's a nucleus.
There's a nucleus. Why is that important?
That's where all of the cellular machinery is.
Good. So what's the cellular machinery? What are we talking about there?
Like the smooth and the rough ER.
Is that in the nucleus or
Apparently not, since you're asking me.
what's ER?
The endoplasmic reticulum,
Okay.
which is in the cytoplasm.
Good. So your endoplasmic reticulum. There's smooth and there's rough. Why is it rough?
It has proteins on it,
Yeah, well,
ribosomes.
Good, good. So you got ribosomes. Why are they important?
They do stuff.
And make proteins.
They make proteins. What do they make proteins from? So I've got a ribosome here that is making a protein and putting it in there. It's using amino acids, right? I'm going to blow this up a little bit on screen. Good. So here's a ribosome. It's going to take amino acids. How does it know what amino acid to put there?
Based on the codon.
Based on the codon on what?
On the RNA.
Good. RNA. Let's do RNA in blue. So we've got an RNA that has codons on it. What letters are in there?
U and A.
U, A,
G.
G,
C.
C.
C. No T?
No G.
No.
Good.
It's DNA.
Okay. How does it know what amino acids to line up with which codon?
There's a sequence of three bases that code for a certain amino acid.
Good. So we'll just pick these three here. How does it know that?
Huh?
How does it know which one's going to code for that? Little computer men in there telling it. Right, so it's going to have these other things that are going to link the amino acid. They're going to look something like this. They're going to have an amino acid linked to it. They're going to have three bases here. How do they have bases? What is this made of?
RNA.
I should have made that in blue. Yeah, what kind of RNA is this?
mRNA.
What kind of RNA is this? I'm just going to try to keep the colors. RNA, protein. Actually, this would be a lipid layer, but. So, what is this?
RNA.
What kind?
mRNA.
This is mRNA, right? What does M stand for?
Messenger.
Messenger, right? Why is it a messenger? Why is it sending a message somewhere?
The message is the protein.
The message is these codons, right? Telling it how to make the protein, what order to put the amino acids in. Where does that message come from?
It's transcribed from the DNA.
Yeah, which is where?
And
In
then
the nucleus.
Good, right? So you've got this messenger RNA that's going to come from your DNA. And your double helix in here, in the nucleus. So what is this thing here?
Is that translational RNA?
Good, tRNA, right? tRNA is, let's blow it up a little bit. I'm still on screen here. It's kind of clover-like, or not exactly, it's got some kind of weird bump here. It has something here that binds to the different amino acids, and there's about 20 of them, 20-something of them, that are used commonly in proteins. But it's RNA, so it has a bunch of different C, T, G, A going all down here, and there are going to be three on the bottom that are going to be what to these three? Complementary, right? So you're going to get those complementary bases to fit that codon, and that's what's going to tell you which amino acid to line up. So these go through this ribosome like a chain. Again. So this one's going to leave. You're going to get another one come in with I'll do the amino acid in green. You're going to have amino acid number 2. And these amino acids are going to get linked together in a chain, right? Do you remember what the chain is like? The amino acids? Just as an aside, the protein is going to have a carbon backbone. It's going to have a carboxyl end and an amine end. I forget which one is which, but just basically a long chain of a whole bunch of carbons with this, and each carbon is going to have a different amino acid base sticking off of it. And some of them are more fatty. Say you want a protein that's going to end up in your, let's do the double membrane here. So we've got a phospholipid bilayer around your cell. If you want a protein that's going to stick through this, part of that protein is going to have to be fatty, so you're going to get like these aromatic amino acids, a bunch of those. If you want part that's going to be up against the fluid or against the water, it's going to be more hydrophilic, then you want it to be, you know, probably smaller or potentially charged amino acids as well. And there's a bunch of them. These amino acids and what order they're in is going to determine the shape of your protein, right? Because once these amino acids, once you get enough of them in a chain, it's going to then go through some changes, conformational changes. It's going to get packaged into whatever shape it is. And that shape, you know, sometimes that happens in the endoplasmic reticulum, and then those proteins either will be just kicked out into the cell, or they're going to be channeled out in these little vesicles to the membrane if the protein's supposed to go there. So this protein will already be in a membrane and gets kicked out here. So what proteins are important in the body?
All of them.
Which ones are important for us? Are there any that we can treat or target?
Tyrosine kinase.
Yes, good. So a tyrosine kinase. What does a tyrosine kinase do?
The transmembrane protein that is involved in signal transduction.
Good, good. And we know that our good example here of a protein like this is KIT, right? So now before we go further, let's see, I'm going to do a little DNA stuff, and then we'll talk about the Hanahan and Weinberg stuff, the 6 original things. So we got to step back here. This DNA, what's going on here? What's important? How does this happen? This step going from DNA to RNA.
It gets opened up to where it's like the 2 strands get
Good. So,
away.
yeah, so your DNA is all bound around each other. At certain times during the cell cycle, it's going to get packaged up so that it can get split. But you're going to have to open it up and do what?
And then the RNA reads one side and like makes a complementary strand to it.
The RNA does, or
Polymerase.
a polymerase, right? So some protein. There's DNA polymerases that make new DNA, and then there are RNA polymerases that are going to make RNA. So your RNA is going to be made by this polymerase just bringing in bases that are going to be complementary, right? So let's talk about the strands. You've got 2 strands on DNA. Are they both important? They are, but they're complementary to each other, right? So what if we totally, I don't think you could do this chemically, but what if we totally took one of those strands away? If you had a bunch of DNA polymerase in there, you could just make a new strand, right? So there's one copy, but it's got a kind of a double. And that's important because when you go to split the cell, you're going to split this fork. I'm going to do a DNA replication on this side. You're going to split this fork, and you're going to have your DNA polymerase, whatever it does at this fork, come in and make new strands of DNA. I just drew that dash because that's the side where it's going the wrong direction, right? It's supposed to go 5' to 3', but the DNA is wound in this direction. The polymerase can only go one way, so it has to work in little segments, Okazaki fragments, I think they're called. Not too important for you guys. But that's real important in terms of the machinery. So the important thing to remember, too, is that this DNA is going to have different parts to it. And what are those parts?
You mean like the exons and the...
Good. So there's going to be, what are exons and introns? What are those?
The exons are the ones that actually get read.
Yeah, so parts of it are going to be specific. What are the parts of DNA that get read? What are those called?
Exons. Genes.
Genes, right? So the genes are going to be made up of a bunch of exons. They're also going to have introns within them. But blow this up a little bit here. If you've got a gene and only, let's go orange here, only certain parts of it are going to be read or important for the protein, the final product, that protein, you're going to have to take those out. And so that's part of what happens here is it doesn't happen in the DNA. Your RNA polymerase makes that new RNA. That's this thing here. And then parts of it are going to get clipped or spliced out so that you end up with an RNA that is the gene with all the introns out. Make sense?
Mhm.
So that kind of covers the whole, what we call the central dogma. It's DNA, RNA, protein. And that's pretty much everything you need to know about the cell in the most general terms. So how does this swing into like the Hanahan and Weinberg things that we talk about? What time is it? 8:50? Okay, good. It's not 7:38. Good. So what are the 6 things that they talk about in the Hanahan and Weinberg article?
So it's talking about what a cell needs to be able to
Become a cancer, right?
become a cancer. It needs to be able to evade apoptosis.
Good. Well, so how does apoptosis work or apoptosis?
There are signals, well, the cell can signal itself to die
Okay.
based on what's going on in the environment or in the cell.
All right, so we've got a normal healthy cell. This cell is making proteins, and those proteins can be polymerases. They can be, membrane-associated proteins so that the cell can communicate with the outside world. Let's actually, for grins and giggles, before we go on to before we do apoptosis, this c-KIT receptor here, the 2 receptors that come together, how does that do anything in the cell? What does it do? Say you have stem cell factor or something like that, it binds to c-KIT.
And then that basically, via like phosphorylation, like sends signals into the cytoplasm and eventually into the nucleus to like alter gene transcription.
Good, so what are those signals for a receptor for the common receptor tyrosine kinase, and KIT falls into one of these? What's the pathway?
You have your GTP.
You have a GTP what?
Huh?
So this does what? This receptor, these 2 receptors come together, they dimerize, and what happens?
Then it binds to the inside, has the GTP signal guy.
That's a step away, or 2 steps away, but there is a GTP switch in there. What happens to the, and what's this protein called? It's a receptor tyrosine kinase, right? What does that mean?
There has to be phosphorylation.
Phosphorylation of tyrosine. What's tyrosine?
Amino acid.
It's amino acid. Good. So back to here. One of the little links on this chain is tyrosine, and when you stick a P on there, and they cross phosphorylate each other, you stick this P on there. Now it's active, and it's going to send a signal in there. Do you guys know the pathway?
It's like the RAS-RAF, that one?
Yep. There's a couple of steps before that. So there's RAS. There's RAF. Oops. Wait, I'm doing this wrong. RAF. Because RAF actually has to be associated underneath the bottom of the membrane. There's a couple things before that. There is a protein called GRIB2.
Mhm.
There's SOS, which stands for
Save our shit.
Save Our Shit. Stupid name.
Oh, son of sevenless?
Son of sevenless, right? So GRIB2, son of sevenless, RAS,
Because you have
RAF.
like hedgehog and frizzled.
Yep, it's one of the weird ones. RAF goes to,
Ooh, hold on. MEK and MAPK.
yeah. So MAP kinase, which is also MEK. MAP kinase, I'm not sure exactly when this goes into the thing, but this will end up then affecting what genes are going to be transcribed, right? So this signal is not just, hey, we've got a molecule up here on the surface, it's going to do something to the cell. That signal's got to get all the way in there. Now,
what's the
Ian,
ligand for the c-KIT?
you guys tell me.
The stem cell factor?
Stem cell factor is the normal ligand, yeah. So,
Where is that on
It's
Ian said something about a GTP,
here?
GTPase. Do you know which link in this chain has something to do with GTP?
That's where the phosphorus comes from.
The phosphorus from here does not come from GTP. It's close, you're off by 1 letter.
ATP.
Good, right? So this needs ATP to take this phosphorus. And just while we're here, how does Palladia work?
Palladia is a competitive inhibitor to ATP. like binding site.
What kind of inhibitor, what kind of molecule is it?
Small.
Small molecule,
Yeah.
right? So it's this little flat disc-like molecule that's small enough so that it can get into the cell, because this happens on the inside, the juxtamembrane region. Actually, no, it happens at the ABC, the ATP binding cassette, the juxtamembrane regions where the mutations happen, the activating, some of the mutations, the activating mutations. That small molecule comes in here and binds where the ATP would bind. Just to kind of draw this out a little bit more. I'm not going to say three-dimensional, two-dimensional. This protein has a little slot in there that the ATP has to actually go into so that the phosphorus gets broken off and attached to that tyrosine. A small molecular inhibitor, like Palladia specifically, will bind in there and block that site. So back to the GTP, there is a GTP molecule that's kind of important. It's one that's really important because of the way it's set up, and therefore it's important in a lot of different cancers. If you look at all those names up there, is there one that you hear about that's mutated in a lot of different tumors in people?
RAS or RAS?
Yeah, RAS is the big one. And RAS is an interesting molecule, and it's a great molecule to have a mutation because it's a GTP switch. The way RAS works, it's a protein. It will bind GTP, cleave off the phosphorus, go to GDP, and then put the phosphorus back on. And depending on what state it's in, if it has GTP or GDP, is going to determine if it's active or not. So it's a switch. And it will, so if you mutate that, it's a very easy target for mutation. If you're going to mutate something like that, you're going to have an easy switch. You can turn it on and off. So it's a good example of probably why the biology determines why this is such an important molecule for human cancers, for cancer in general. Probably in animals too. We just haven't investigated it as much. All right, back to apoptosis. So the first one of the Hanahan and Weinberg principles that we're going to talk about is apoptosis. How does that happen? Where does that happen? Just keep green for lipids. I mean, there are a couple of different ways that apoptosis can happen. What's the main route?
Usually, like the cell detects an abnormality, and like it has those like death signals that causes it to undergo apoptosis.
Okay, well, what's the effector though? Where does it all kind of come together? You can have death signals from outside, right? There's FAD domain, the FAS-associated death domain. You can have that coming from outside the cell totally, and that actually may be important in some radiation-induced apoptosis. But you also have a lot more internal, right? Because this is mostly internal signals from the cell, where if you radiate your DNA and cause a break, a double-strand break, that cell may want to undergo apoptosis so it doesn't mutate or cause problems. But those signals, and this signal you guys should be somewhat familiar with, this is the ATM, ataxia telangiectasia mutated. I know I mentioned it a few times. But what's more important about it is that protein, say it finds a I'm going to make a double-strand break. Say it binds to this and identifies a double-strand break just because of the conformation, it's going to go tell another protein, go tell mommy, it's going to tell another protein that something's wrong here. And what's this protein? Huge, important protein.
P53.
Good. ATM, there are other steps involved. They can go other routes, but it directly binds to P53 and activates it.
ATM directly binds to P53.
Yeah, and this is just one pathway where DNA damage can be recognized. But where does the effect happen? Like what tells the cell, hey, it's time to just die? I mean, P53 can do a couple of things. It's a gatekeeper. It can just shut down the cell cycle, right? We know that it can be directly involved in that. But it also can say, hey, we're done here. Just shut down the whole factory. Let's kill this cell. But what organelle does that happen in?
Mitochondria?
Good. So whatever apoptosis signals you're going to have are going to get out to the mitochondrion, or they could start there originally. And this is the classic pathway for apoptosis. And it's going to cause some molecule to leak out of the mitochondrion.
Cytochrome C.
Good.
That's down here, Mickey D. Oh, fabulous.
Should be on there somewhere, yeah.
not in there, but at least that part is.
But it's really important
Yes.
because it's important for radiation, so there you go. No, ATM is just one example. Probably the more important part of that is the P53 side of things.
And that's up here.
So there are a bunch of pro and anti-apoptotic factors, right? Do you know any of them? Those would be proteins.
Bax and bad are apoptotic, they have A in them. And then BCL is anti-apoptotic because it doesn't have A in it.
BCL2, right?
Yes.
Yeah.
Let's have
And
that.
there's other BCL, XL. If I don't know, you should probably have a little table of that you look at
Yes.
shortly before
And
boards.
if they don't have A, they're anti-apoptotic, that's how I remember. Bax and bad are apoptotic.
So evading apoptosis, that's number one. I'm gonna put that over here. Well, it's not number one, but it's the first one we picked. How about other ones?
It can grow without like being signaled to do so.
Good.
Like, it can grow on its own.
So what do they call that in the paper? It's
Self-sufficiency in growth signals.
good. I'm gonna abbreviate that a lot. Self-sufficiency in growth signals, SSGS. So do we have an example of that?
The CK mutation.
It's all there, right? So you have a dog with a mast cell in the skin, and something happens, you get this here, probably one of the more common ones is it gets a juxtamembrane mutation that causes this protein to be constantly active or turned on. Now you've got growth signals pouring in. Now where does that happen? Where does that mutation happen?
Exon 8.
What's that?
Exon 8.
Exon 8 or
11.
11, the two ones, where are those?
Juxtamembrane.
There, yeah, but does the mutation happen here in the juxtamembrane region? The protein's mutated, but where does that mutation happen?
In the DNA.
Good. Yeah. So back where this DNA codon is, you're going to get some kind of mutation. Usually, we'll start with a point mutation. There are lots of different mutations, but a point mutation would be a common one that we see. That's going to be in the codon that codes for some protein in this part of the molecule. All right? So you've got to follow it all the way around. So that's number two. How about others from there? You guys can cheat.
Insensitivity to anti-growth, basically meaning it doesn't, when it gets the signal for apoptosis, it like ignores it or doesn't respond to it.
Yes, but it doesn't have to be just through apoptosis. That's good. I mean, apoptosis, evading apoptosis is important. But you can also, this is a growth signal from outside. You can have negative growth signals from outside, right? What happens to normal cells when you plate them in a petri dish?
Oh, the cell-to-cell contact inhibition.
They grow until they hit each other, right? And so there's proteins out here like, I'm not even sure exactly which ones are CAMs, integrins, things like that are going to bind either cells to each other, cells to the endothelium. But if you were to plate these in a petri dish and put them there, they grow until they hit each other and then they're going to stop. If you take a cancer cell that's lost this whatever inhibitory signal, the X on it, that's going to tell the cell stop dividing when I touch something, then they're just going to pile on top of each other and the petri dish will just get covered over. And that's one of the things that they use when they're, and they still do this a lot, when you're looking to see if a cell that you have in culture is a cancerous cell, you'll do this. You'll plate it out on a dish and see, that's one of the things is do they grow on top of each other. So that's important. What else do we have? So that was the insensitivity to growth signals, the anti-growth. What else?
I don't know what the difference is, but one of them is limitless the limitless replicative potential.
Good.
It's like when the telomere
Good, right?
gets eaten away.
What are telomeres?
The little caps on the DNA.
Yeah.
Good. Little caps on the DNA.
And they eventually they wear away as the DNA divides.
Good.
And then once it's gone, that DNA will that cell will be undergoing apoptosis. But then the cancerous cells, they either add to the telomere or prevent it from wearing away so that it can just keep.
Good. And it doesn't necessarily have to be apoptosis. There's another word for that. When your telomeres get short and the cell decides I don't want to divide anymore, what do they call that?
Senescence?
Senescence, right? So if they were to take a biopsy from me right now, my normal skin, assuming I don't have skin cancer, and they were to take some fibroblasts from there, throw them in a petri dish, and try to grow them, they might grow for a while. You might be able to get them, if you put them in with the right growth medium and the right signals, they might grow for a while. But they would get to about 15 to 18 or 20 cycles, and then they would stop growing because they're programmed to stop growing at some point. This is why some billionaire in Texas is dumping a bunch of money into research. He wants to try to figure out how to extend telomeres so that he can live forever. So this is part of the normal signal. It's part of the reason we die right now. If we can figure that out, we'll live forever. I don't know about quality of life, but we can potentially live forever. But cancer cells can use that, and they have, what was the enzyme we said? Telomerase, right? So telomerase will extend those telomeres after they've been taken off. And why do they get shorter?
Every time you replicate, where the polymerase grabs on, it has less and less to grab on.
Yeah. When you replicate, you go from one end of the DNA to the other. It's not going to start right at the very end, so you're going to chop off these telomeres bit by bit as you replicate each time. So, and it's important. I mean, all of these things that we've seen here are important for normal cells. This one's important because these cells have to divide a lot when you're a fetus or an embryo and you're growing, or you're a child. But when you get to be an adult, if it kept growing, we'd all grow till we're 30 feet tall, and then we'd die from back or foot problems or heart disease. So you have to have these things in there in order for the body to grow to its natural size. And apoptosis is an important part in development, right? Where does that happen? There's an example of apoptosis right here in this hospital.
The tadpole.
The tadpole, right? What happens with the tadpole?
Tail falls off.
Yeah, his tail's going to fall off. Does it?
Yeah, just what is it?
That's an iguana.
Oh, but I thought he eats his tail.
Do they eat their tail?
I don't
Yeah,
think
because
they
Becky
eat
said
their tail.
she stops feeding it, and his tail falls off, and he eats his tail.
Parts of it may fall off, but a big part of it is apoptosis. The tail just dies. I guess I don't think it falls off though. It just
I
kind
don't
of
think they
shrivels
eat
up. Yeah, the body absorbs it.
Maybe that's why hers don't
different.
ever live. She says you're
He
supposed
stops feeding it.
No,
So
not
he-
yet.
Yeah, you stop eating it, and it eats its tail.
I think if you
and
keep
falls
Starting
off.
to
feeding
use
it,
back legs.
it will still fall. It will still shrivel up and go away. Where does it happen in people in development?
In our tails.
Our tails. We do put tails in embryo. Yeah. What else? Where else?
Our little finger webbing.
Yeah, finger webs, right? My dad had webbed toes. Just
Really?
his two middle toes had webs between them, so it's some
That's
kind
Like
of
from
apoptosis defect.
after like a little web.
Like 3/4 of the way out. They were just like fused together. I assume that's some kind of defect in apoptosis in his toes. I don't know. Never looked into it beyond that.
Was it on both feet?
Yeah.
Huh.
Yeah. So anyway, if we didn't have apoptosis, we would all have big clubs for hands. We'd have the bones in there and everything, but we'd have a big thing of skin going all the way. Like, be good for handball, I guess. But for swimming, yeah. My kids wear those stupid paddles when they swim too. It looks like they've got webbed hands. Where were we? So we're on senescence, and that was
Limitless replicated.
limitless replicative potential. So what's next?
Angiogenesis.
Angiogenesis. So not so much just in this cell, but how does angiogenesis play a role? Now we're going outside the cell.
It makes its own blood supply so that it can keep growing.
Good, right? Yeah, cancer can't survive without nutrients. The cells can't survive. So in order to do that, they have to cause blood vessels to pop up to give them a good blood supply. How big can a cancer get before?
2 millimeters.
Good, only about 2 millimeters before it needs a new blood supply. What's an example of a molecule involved in this? Right, the most common one.
VEGF.
VEGF, right? Good. So where would that play a role potentially?
Like we're in the cell?
In or outside the cell? Like where's the VEGF coming from? Where's it going? What's it having an effect on?
It's coming from in the cell and then goes to blood vessels.
Good. So a cancer cell might, for whatever mutation reason, might make more VEGF. I guess now would be a good time to talk about mutations. So I've got a cancer cell that's making too much VEGF and it's kicking it out of the cell. Why might that happen? Go back up here. Let's say here's our VEGF gene. We got one copy of the gene. So what could happen to make this cancer cell do something different there?
If the gene is constitutively activated?
Okay, so the gene is constitutively activated. So VEGF is just a protein that gets kicked out. It's not like a molecule that needs to be activated. So how would you do that for a protein? Here we got an obvious thing. We just change the shape of this and it's turned on. Here we've got a switch. If I screw something up in, where the GTP binds, I can turn on that switch all the time. Here we've got a molecule, and the cancer cell wants to kick out this normal molecule. How might it do that? So how's it going to make it? It's not doing anything different to the protein, so it's not constitutively active, but it's more, what else can happen?
Upregulated.
What's that?
Upregulated.
Yeah, how do you upregulate something like that?
So it's hypoxic,
You'd have
and
to-
maybe it would kick out more.
Okay, so hypoxia can do it as a natural process, right? You know what molecule's involved in that?
HIF.
Good, HIF,
One.
more, yeah. So HIF1,
Alpha.
alpha, there's other parts to it too. But that, if that gets activated because of hypoxia, that can cause more VEGF. And that can happen in our own bodies, right? If we have parts of our body that become hypoxic, I don't even know, that may even happen to some degree with higher altitude training and stuff like that. But if we have hypoxic areas, it's going to cause more VEGF to be kicked off. That's normal. How can a cancer cell bypass the normal here? Well, what do we want it to do with the VEGF? We want it to...
We want more of it.
We want more of it, right? How do you get more of it?
You make more of it?
You make more of it, right? So we've got a gene here. Whatever's controlling this gene, it's, what do they call that? How much a gene is making itself? It's expression, right? So whatever's controlling the expression of this gene, usually there are these upstream regulating parts, could have a mutation in it, telling RNA polymerase, just keep making copies of this. Just keep making copies. We're just going to kick out as much VEGF as possible. So that's one way. What else could you do? I mean, obviously there could be some down-regulating signals telling VEGF to stop, like things that control HIF1, if you lost those. But what else could you do from a mutation standpoint in the DNA?
Ooh, a little upstream promoter guy can just always be on.
Well, that's this, right? Yeah, so you've got an upstream promoter. If there's a mutation there that tells it keep making VEGF, keep making VEGF. But what if you have a normal upstream promoter? What else could you do in a cancer like this? Well, you want more VEGF, maybe you get more VEGF genes, right? And this happens in cancer cells, probably more towards when they're becoming more, they're getting more chromosomal instability. But you could have, instead of one copy, you could have 10 copies of VEGF in there, all with normal promoters, and you're just kicking out 10 times as much VEGF, just because they're all there, they've all got that same signal. So anytime you get a hypoxic signal that tells you to make more VEGF, instead of making one molecule, it makes 10. Instead of making, 1,000 molecules, it makes 10,000. So that's another type of mutation would be an insertion of some kind of gene. So when you think about all this stuff, before we move on to the diagnostic testing stuff, think about that is, well, how can I screw this up? I can either make more of something or make something more active, or I can make something less active or get rid of it, right? So if I've got a protein that, say my ATM or my P53 here, that tells a cell to stop, how could I change that regulation to make a cancer cell?
Well, you mutate it so that it doesn't get that signal.
Right, you can mutate it. So say now we've got say somewhere else, we've got the P53 gene. This should be all in the nucleus, I'm cheating here. Let's say we've got a P53 gene somewhere on that chromosome. Actually, I'll keep it in the nucleus so we're not. So we're going to call this one, this is our P53 gene. What could we do to it?
Or if it's downregulated.
Good, so yeah, we've got promoters that tell it when to make new P53. So we could squash that, right? We could cause a mutation in the promoter. What else could we do? What about the gene itself?
Could not be there.
It could not be there, so you could have a deletion, right? Part of the DNA is gone, and you've got no P53 gene. If you're a cancer cell, that would be a good thing for you. What else could you do? and this is really important for you guys to understand, is this whole thing where you go from bases, get transcribed into your RNA, gets spliced, gets put out to the ribosomes, and gets coded, one little screw up here can mess it up, right? So what else could you have? If you have one little screw up in this P53 gene, one base pair is wrong, then you might activate it, you might turn it up, but if you're a cancer cell, you might deactivate it. One little base is off, and this protein folding might go from this to, might go from alpha helix to beta pleated sheet, and then your protein is a completely different shape. It could be garbage, or could be doing something completely different. But one little base. Now there is redundancy in this DNA code, so one little base may not do that, right? UAG might code for the same thing as UAC or UAA. So it doesn't always do it. Now, how often does this happen in cells?
What?
A little mutation like that.
All the time.
Yeah, it probably happened hundreds of thousands of times since we sat down here this morning, right? So the body takes care of that in most cases. It has repair mechanisms to find those changes and fix them. But if those repair mechanisms are screwed up, or if you're unlucky, it's part of getting cancer is you have bad luck. If that mutation happens to be in P53 and stay there, the body can't fix that mutation. Now you've got no P53 in your cell, in that one cell. And that cell's just going to keep growing. It's not going to undergo apoptosis. It's going to keep growing, and that's how you get a cancer. Make sense? All right. Let's wrap up with the last. We did angiogenesis. What else we got for?
Tissue invasion and metastasis.
Okay, good. So yeah, all this is great to make a ball of cells somewhere, but a ball of cells somewhere in your body is not going to kill you, right? So it has to get a blood supply so it can grow big enough, but then it's got to go somewhere. It has to invade into tissue so it'll kill you. It has to grow into bone, make it so that you can't eat, or spread to your lungs, spread to your liver, and start to damage those tissues. So how does that happen? What are examples of that?
And that's like the where it ends with an M, like it opens up and makes the vessel so that it can go and spread.
So there are proteins that can be given off.
Yes.
There are MMPs.
That's the word.
And tissue inhibitors of MMPs.
Mm-hmm.
Metalloproteinases.
Yes.
So these are things that are involved in breaking down proteins. And if you're a cancer cell, at some point you're going to want to do that so that you can grow through the tissue, get to the blood vessel, get to the brain, whatever you want to do if you're an evil cancer cell. So that's a good overview, I think, of the general parts of it. Now, how do we test for different stuff here? So the testing in here, I'm going to kind of go through it like they do in the chapter. We'll do it pretty quick. The first test that they talk about are cytogenetics and karyotyping. What is that used for?
To look to see if there's it's like when they fluoresce and there's like multiple colors on one chromosome, then you know there's like been the translocations.
Okay, good. So you could find a translocation. What are you actually physically looking at, or how do you do that?
You're looking at like the metastasis spread or something.
Metaphase.
Yeah, that's
Right?
Mm-hmm.
Not a rabbit. Long camera. Oh no, battery's dying. All right. So you're looking at a metaphase spread. You're looking at the chromosomes all split up. Give me 1 second.
We're gonna be fine. I do want to go through West at some point too. Yeah.
All right, still going. So we're looking at this, and you're going to look for some kind of translocation, right? So this, what is it? It usually has bands on there. They're called G bands. You know why? Dark and light bands. Giemsa is actually the stain they use for that. So those G bands might look different. So if it normally looks like this, you might have a part that has a different characteristic with the G bands here. How sensitive is that?
Seems pretty sensitive.
It could be sensitive for a big translocation. Is that going to find this mutation here?
Probably not.
No, right? Because you can have a point mutation in here would not change this band at all because it's only one base in your DNA. So this was one of the original tests that was used a lot because it's all they could do back then. They didn't have any of this molecular testing. So what they relied on were major changes in your chromosomes. What other changes do we see in our chromosomes? So these are translocations. There are other changes. What are some examples of this in cancer?
Philip.
BCR-ABL thing.
Good, good. There's two good examples. So that's, what's that one called?
The Philadelphia.
So there's the Philadelphia chromosome, and there's another one for another lymphoma. You guys should know these. But yeah, they're major chunks of the gene. It just happens where that translocation happens causes a fusion gene of BCR-ABL that makes an active protein that's going to cause proliferation, I think. But more importantly is to understand this test. If you take these cells in metaphase, you stain them, and you plop them out like that, you're looking for massive changes in your DNA, which you could have the worst cancer in the world, and it might have a totally normal karyotype if it's got a bunch of little point mutations. Now, why do you usually not have that? If you take cancer cells and you biopsy them and you look at these metaphase spreads like this, why can you usually find changes? Usually they have all kinds of changes. They'll have different numbers of chromosomes. They'll have freaky looking chromosomes. Why is that?
Because don't you have like not just the translocation, but when you have like the double centromere thing, they can look weird?
That's more from radiation damage, but that can happen in cancer cells as well. We talked about the dicentric chromosomes. But what happens as cancers get worse and worse, they undergo chromosomal instability, right? So in order for a cancer to really take off, one mutation like this in P53 is going to tell your cell to go through the cell cycle, and it's going to tell it not to undergo apoptosis. But that doesn't necessarily mean it's just going to be a cancer, right? How many cancers have just one mutation?
Not many.
Not many, right? RB is one that we know of, retinoblastoma. But most cancers require multiple mutations. And as you go further, all those mutations are going to be in things that are going to control the chromosome. They're going to control the expression, and they're going to control the things that are watching this chromosome. So once you start to lose all of those, you're going to get chromosomal instability. So in cancer cells, you will get lots of different mutations. It's an old paper, Kevin Hahn did it, but he looked at metaphase spreads in dogs with lymphoma. He found that trisomy 13, so 3 copies of chromosome 13, was prognostic for dogs with lymphoma. Could have been a statistical fluke. Could have been there's some gene on there that's now overexpressed, because if you've got 3 copies of this chromosome, you've probably got excess copies of that DNA, right? It's the same thing as putting in these multiple copies of, say, VEGF. If you've got a whole chromosome full, some of those might be genes that might tell your cell to divide and invade and avoid apoptosis and things like that. So that trisomy 13 is something that really wasn't followed up on, but it's in there in the literature. And there are other molecular analyses that are being done based on that. So what do we want to do if we want to be more specific? And as you go through chapter 2 or chapter 4 of the old book, most of the tests are based on this principle. So if you want to get more specific and look at the DNA, you want to look at certain parts of it, what tool do we have?
Microarrays.
Yeah, microarrays are one test that uses this. But you want to, so we don't, it's great if we can see the chromosomes are screwed up, but even that doesn't tell us that much. If we look at this, it doesn't tell us what gene. They just happen to find this BCR-ABL because they investigated it. But you could have, the funkiest looking chromosome with a long arm. It doesn't tell you what genes are involved. So now we got to find the specific genes, or we got to find the specific mutations. So what principle or what thing are we going to use to find that? And it's also how do these DNA strands stay together?
They have hydrogen bonds,
Hydrogen bonds between what?
the bases.
Yep, in particular, they're complementary bases, right?
Mhm.
The bases match up with each other, G and C, T and A, U and A, if you're talking about mRNA. So the reason why this stays together so well is think about it like a huge zipper, right? A zipper in and of itself can hold stuff together, or Velcro can hold stuff together because there's lots of little connections there, right? if you were to take one little hook and eye from a zipper and try to hold your pants together, it wouldn't work. But you've got 100 of them in a row, it's going to keep your dress or your pants on. With DNA, I'm going to go outside. I'm going to make my nucleus bigger. We're going to go bigger nucleus here. With DNA, I'm going to open it up even though it's not open in real life. If we want to find, say, a small mutation here, we can take advantage of this process. It's called hybridization, right? So you can take either other DNA or MR or RNA, doesn't have to be mRNA, it's RNA, with the complementary bases, and if you've got a match, and it doesn't have to be a long match, it can be 10 base pairs, it's going to undergo hybridization and it's going to stick there. So what does that tell us? Well, that tells us, say we know this mutation that exists, say we know we're missing a certain a certain base in P53 and it's deactivating it. Well, if we know that, we could have that sequence on, say, an RNA, and we can tag it with some kind of fluorescent marker or radioactive marker. Usually now they use fluorescent markers. So, and if we do that in situ, that's called fluorescent in situ hybridization. And it can be used here, and the reason why it's in situ is because it can tell us where in the cell it's going on. If we do this, we dump these fluorescent markers on the cell, it'll bind to where it's happening in the nucleus or in the cytoplasm. There's DNA roaming around. You can do it on chromosomes, right? If you want to find a particular gene on a chromosome, you get that complementary base pair. Say you know that there's a string of bases in the mRNA that codes for this BCR-ABL mutation, the point where the two proteins come together. If you find a primer or probe that can hybridize to that, tag it with a fluorescent marker, you put it there, it'll light up that chromosome. You also have markers for chromosomes, and that's the chromosome painting, right? You hybridize markers that are specific to, that are for genes or combinations of base pairs that you'll only see on chromosome 3. Chromosome 3 is going to turn whatever color. It's a false color they do with computer generation, but you can turn that chromosome green or pink or whatever color you want. And there's a nice picture of that, I think, in both versions of Tannick and Hill. So hybridization is the key for all of these other tests. Now, the other test that probably is the other bigger part of this whole thing was what? What's the main test we talk about with cancer? The thing that kind of revolutionized this. Because this is great if you know or you're looking at certain things, but we're talking about a single base pair in one strand of DNA in a number of cells. How do we get more sensitive? What test do we use for that?
PCR.
Good, right? How does that work?
So you make lots of copies of your DNA.
Good. Just any DNA?
Of the piece of DNA that you're interested in.
Good. How do you know you're going to get that piece?
You need to have the primer.
Primers, right? So you've got a piece of DNA and you want to know, is this gene in my sample, right? So say I've got a cancer cell and I want to know, well, does that cancer cell actually have any P53 gene left? You could take primers that you know are going to be in that P53, put them on there, hybridize it. Now, why is PCR different? This we could do. We already could do this. We just take a primer and put a fluorescent probe on it. Boom, you're done. What's the PCR part about?
There's a chain reaction.
Yeah. What does a chain reaction do? It's not good if you're working at a nuclear plant, right? Because what is it?
It just keeps going on and on and on.
Yeah, it's like a vicious cycle. It usually multiplies. So how does that work with PCR?
You get lots of copies of just that one section.
How?
Because you have the primer or the polymerase.
You got a primer, you got a polymerase, right? So you've got a section of DNA that you want to make copies of. So you put your primers in on either side, flanking primers, and you put in a bunch of bases and they'll hybridize, right? Then what do you do? So now it's stuck to the other side.
No, you replicate them.
I already replicated this piece, right? I put in polymerase and I made this strand, so I got a copy and it says, yes, the P53 gene is there, but how the hell are we going to find one copy? We're not, right? We've got to increase the number of copies, right? So how does that happen?
So you do that by...
How do you denature or separate the strands on DNA?
Oh, it cycles through the temperature.
Good, right? You heat it up to, it's like 93 degrees or something like that. These strands are going to come apart. You've got your original strand and you've got your new strand that you made. You cool it down, you've still got your polymerase and you've still got your bases in there. You've still got your primers in there. Now you guys have 2 copies. So this is why it's a chain reaction, is because now it becomes exponential. You guys play 2048? Game's addicting. You played it? Don't download it. So it's exponential. Now you've got, well, you had an original one, now you've got 2 different full sets of copies here, then you got 4, then you got 8, then you got 16. So you can make large amounts of DNA, and the way it's a cool test too is that they've automated it, so they have a polymerase that doesn't break down when you heat it to 93 degrees. So they throw all this stuff into a test tube, you put it in a machine that just cycles the temperature, and you come back an hour later and you've got millions and millions of copies, or however many copies you think you need of that. So it's an extremely sensitive test if you're looking for whether this is present. Now you don't have to necessarily do it for just a gene. If you're using primers here, what you're testing for is, do these primers have something to bind to? So say you were looking, and this is where the sensitivity comes in, say you were looking for, here's your P53 gene, and we've got a point mutation here. If you know where that point mutation is, you could set up a primer that has that same, the complementary base to that thing, put it in there. If this primer sticks and you have to have the other primer here, you make more DNA, that's a positive test for this mutation. So when you're testing, using PCR to test for specific mutations, if you know them, this is often what you would do. Now we got to take a step back because we got to cover the general stuff. We jumped to PCR, but we talked about DNA, RNA, and proteins, and you guys should know this. How do we test for these?
Northern blot, southern blot, and western blot.
Good, so which one's which?
Southern.
Southern, so start with Southern because that was the guy's name, so that's DNA, and then
Northern is for RNA.
RNA.
And
Good.
western is protein.
So what's going on with those plots?
So you cut, I have it all right here.
That's fine.
You cut your DNA into defined lengths using a restriction enzyme.
Good, good. So what restriction enzymes are what?
They,
Where do we get them from?
I don't know, but they cut things in a defined
Bacteria.
spot.
Yeah, good. They're actually used by bacteria to kill plasmid DNA. That's where we found. Now I think we make them in a lab, but that's where we found them. Is that they would cut DNA at very specific points, so that they don't cut their own DNA. So these restriction endonucleases. So we've got a mush of DNA in a vial. You'll cut it with these restriction enzymes into different pieces of DNA. Then what do you do with it?
Run it through a gel.
Run it through a gel. What does running stuff through a gel do?
So
It
things
separates
that
it by
are-
size.
How do you run it? Just say go run.
With
By
an
electricity.
electron.
Good.
Electrons.
Why does it?
Because DNA is electrically charged, and so it's going to move towards the positive end.
Good. It's got a negative charge. The phosphate backbone, phosphorus has a lot of negative charge associated with it, so that phosphorus backbone is going to be highly negative charged. Now, why, if we put it in a gel, and you guys have all seen these, we put it in the wells here, you're going to get bands at different spots, right? Why?
The gel is like a matrix, and so the larger molecules are going to be inhibited. or
Good.
retarded.
Yeah. So the gel, it's got to go through the gel. So large molecules aren't going to get very far. Smaller molecules are going to get further. And so you could compare this if you had 2 different, say you had DNA from a normal dog and DNA from a dog with some kind of deletion. You might be missing one of these big pieces missing. You might have a band in the normal dog and no band here. Or say you've got, it would change. Something would be different with it. So you can have too many bands or not enough bands. And so being able to kind of look at that and interpret that, comparing a normal or a wild type versus some kind of mutant is important. RNA, basically, same thing, right? How about for protein? The Western blot. It's basically the same thing, right? All these proteins, these side chains on these amino acids can have different charges. So different proteins will be different sizes and have different charges. So they'll move through the gel differently, but you're able to look at them that way. If we have time, we'll cover it in a few more minutes. So we talked about fish, we talked about the PCR. They talk about comparative genomic hybridization. That's kind of taking this stuff where you take your restriction enzymes and split it up and breaking it up for the whole genome for something, putting it into a library so that you can use it for other testing. Let's see what else we did. Okay, single nucleotide polymorphisms, what are those?
Oh, SNPs.
With
SNPs.
the DNA differs at a single.
Yeah, so that, I mean, we talk about mutations because these are usually things that cause big effects on these proteins. But you can have changes in your DNA that don't always just shut down a protein or activate a protein. You can have polymorphisms, right? If we all had the exact same DNA, we would look different. And polymorphisms are things that you can use to identify differences between people or differences between genes and stuff like that. So it's using these same principles. You can find it the same way. You can do a fish to look for a specific mutation in there. If it changes the size of a gene, you could look for it that way as well. We didn't really talk about epigenetics, but how does epigenetics play a role in this?
That's more of how the genes are expressed, like not so much. That looks more about like the gene expression versus like the actual.
Yeah, so we talked about